Good evening. Today, I'm Nur Hamiza binti Muhammad Shah, third year student in Genetic A163175. I'm going to talk about a research article from the BMC Genomic. Title: Limitations of Variable Number of Tandem Repeat Typing Identified Through Whole Genome Sequencing of Mycobacterium avium subspecies paratuberculosis on a national and herd level. Mycobacterium avium subspecies paratuberculosis is the bacteria which causes the jaundice disease. This jaundice disease is a type of inflammatory bowel disease in cows. This bacteria not only give bad effect towards the cattle health, but also affect the economics of dairy production system. Besides, this MAP bacteria also can cause the crown disease in human. In this experiment, the variable number of tandem repeat and whole genome sequencing methods are used to investigate the MAP incursions. The aims of this experiment is to assess the diversity of MAP isolates type 2 in Canadian dairy herds using the whole genome sequencing. Also, to assess the ability of Vntr typing to identify unrelated and related isolates at the national and herd level. The first method is MAP isolates and DNA preparation, which the samples are collected from the 6 different participating regions of dairy herds, which are stated as a Table 1. The second step is Vntr typing, which are assigned according to the MAC INMV database. The third method is whole genome sequencing. This whole genome sequencing map DNA was prepared using the Nextera XT sample. The last step, which is data analysis. This data analysis uses the cond tree to trim the raw rates, and it is mapped to a revised versions of the K10 reference genome using BWA. The variant sites are identified using the SAM tools and are filtered based on the coverage, mapping quality, and heterozygosity. The data from the SAM tools are visualized in tablets to investigate the specific polymorphisms of interest, and from there the phylogenetic tree was created using the field mill. From the results of the Vntr typing, you can refer to the Table 2, which showing the number of map isolates, herds, and whole genome sequence within each Vntr type. From here, you can see that the INMV type 2 belongs to 76% of map isolates. The INMV type 3 belongs to 8% of map isolates, while the INMV type 1 belongs to 7% of map isolates. The loci 3 and 32, which are not polymorphic, while the others are contain 3 and 2 alleles. The INMV68 belongs to map isolates bison types, which are identified in 7 herds. From the whole genome sequencing method, 124 isolates were successfully sequenced, representing 11 different Vntr types, with an average coverage of 33x. From Figure 1, it's showing the maximum livelihood phylogenetic tree created from the 3039 high-quality variant sites that were identified in WGS. The analysis of Vntr typing and the whole genome sequencing. In this dataset, the map isolates differing by less than 10 SNPs belong to different Vntr types 10% of the time. So this value increases as the number of SNP differences increase. The figure 3 shows that the INMV3 isolates were on average more closely related, with less than 50 SNP differences in the largest cluster, while for the INMV2 isolates, which were approximately 100 SNPs different. Meanwhile, for the INMV1 isolates had on average over 150 SNP differences between isolates within that type, with one isolate exceeding a differences of 200 SNPs. The table 3 showing the absolute minimum and maximum pairwise SNP differences within and between the three major Vntr types. From the figure 4 is showing the locus specific changes in the repeat length, where the locus 10 had the most instance of convergent evolutionary, and locus 47 and H3 are relatively stable. From the discussion, the genetic relationship of 124 Canadian bovine map isolates was assessed using the WGS and Vntr typing. Few findings has been identified from this analysis. It shows that 8 genetically distinct map subtypes exist in Canada, with over 80% of isolates belonging to a single dominant subtype. The Vntr typing may cause the overestimate and underestimate relatedness. The conversion evolutionary was observed. The phylogenetic tree of map isolates are created based on the 3039 SNPs identified through the WGS, was analyzed, and 8 divergent subtypes were revealed. The analysis of 3 herd-level dataset illustrates the value of using WGS data for the study of epidemiologically linked isolates, and that Vntr typing data may lead to incorrect assessment of diversity and relatedness of strains. From the current study suggests that the current Vntr typing scheme includes the loci that are too unstable and therefore unreliable to be used for the molecular epidemiological analysis of map at both broad and limited spatial scales. In the conclusion, caution should be used when using the Vntr typing as a tool to assess the diversity and relatedness of map isolates at both a national and herd level. The whole genome sequencing provides unparalleled detail regarding the genetic profiles of map. Thank you.